Well, given that we are all gathered here and the recording has begun, I say let's start. So, welcome everyone to the next in our Astrobiology webinar series. Just a few very short announcements before we begin the presentation. The slides for this presentation are already on the website under the event for today. So if you want to look at them afterwards, or if you're only listening to us on a telephone link, you can go and download them now. The event is being recorded, just so you're aware. And we won't be having any presentations next week. So in fact, just check the event list and you'll see we start again on the 8th. But we do have a presentation tomorrow. Okay, and with that, Irene.
Okay, thanks, Andy. So as everyone here probably knows, we are talking about the evolutionary pathways and emergence in molecular systems concept document, which was written by three of us during the Wallop session, myself, Michael Tice, and Aaron Goldman. So I am basically going to just go over the current document, and then we're going to have additional input from different sides, Eric and Andrew, and then we'd like an open discussion for people to add their comments. So I see the goal for today as being trying to fill in the gaps that we've created in our concept document. So during the road mapping process, I think that all these individuals came together and described things that they thought about, and their individual thoughts kind of form the roots of this tree. And eventually, we want to fill in the gaps to the extent that we can help the people who are making the strategy document make an inclusive document that will be flexible enough for people even who haven't been represented here. And then the science hopefully will flourish from that. So I think your comments, I'm really hoping your comments will help us fill in the gaps in the current document. Okay, so we want to improve the document, and you can do that by making comments now, and then the most important thing maybe is to record those comments directly onto the online document after the webinar. Okay, so this concept document was intended to capture sort of system-style studies and to emphasize the analysis of dynamics. There have been some other concept documents that focused more on the synthetic biology and the, I don't know, maybe the biochemistry of it, and we were hoping that this document would kind of capture those, I don't know if you want to call them higher-level types of ways of thinking about these systems. So the summary is that life is an evolving system of interacting molecules. So the evolution and the interactions are important things to think about. With regards to the evolution, we want to understand the capacity of chemical systems to evolve greater genetic and metabolic complexity. And this is all a little bit vague, but maybe intentionally so that people can add their own thoughts in here. And also to understand evolvability, evolution of new functions, and evolutionary pathways that would be available to these first organisms. On the interactions side, the thinking is that molecules aid each other's fitness through adjusting each other's stability or replication or self-assembly, et cetera. And network organization and cooperative cycles could allow the emergence of these systems-level behaviors at the level of a cell or a cycle or higher than that, which are beyond the capability of individual molecules. And so we want to understand those networks of interactions. So the specific sub-questions that we wrote down, and this is not an exhaustive list, but it's just what's in the document now, are trying to understand the structure of networks, how do or did individuals emerge, how does the network structure of molecular or organismal interactions affect evolution, and what network architectures are kind of better in supporting survival and evolution. The model systems, and this overlaps quite a bit with some of the other concept documents. What are, or how do physical and chemical properties of biopolymers affect their evolution and evolvability? Can we build experimental systems of molecular networks and cycles? And that definitely overlaps with some of the other documents out there. What can we learn about the evolution of life from long-term experiments on the evolution of microbes? What can we learn about the evolution of early life from artificial life? And that would, I hope, bring in some of the computer scientists who have thought about this type of thing. And one of the comments that I think Andrew Ellington made in the concept document is this brings up the question of what is an individual? And at least Eric is going to elaborate on that question later. The next heading that we have is evolutionary landscapes. In other words, pathways for evolution, and walks through the space of possible sequences. How reproducible is evolution? How diverse are the evolutionary pathways under same conditions or different conditions? How does evolutionary history constrain future pathways? Basic questions about what evolutionary landscapes look like. Are there neutral networks? Are there lots of evolutionary pathways that can lead to the same results? How do evolutionary landscapes and sequence space translate into landscapes in structure and function? What's the mapping there? In trying to understand the idea of evolvability, how do evolutionary innovations come about? What are advantages and disadvantages of modularity, which has been talked about quite a bit in understanding systems? What are good metrics for evolvability of different chemical systems? And this is an important point for what exactly are we talking about? How do evolvable chemical systems arise from non-evolvable chemical systems? And that, again, overlaps with some of these synthetic biology documents. And are there characteristic timescales of evolution? An important variable in all this is the environment, since fitness depends for sure on the environmental context. So how do environmental conditions affect and constrain evolutionary pathways? How do prior genetic context and history affect and constrain future evolution and emergence? Here's a list of so that's what's in the document, and here's a list of the connections to other concept documents. You can look at this list, and I think that there are, certainly overlaps, but this document was intended to bring out more of the kind of, analytical and systems biological view, I guess. Okay, so now I'm going to hand it over to Eric, and Eric is going to talk about a more abstract side, and then Andrew is going to bring it all together, and then we'll have an open discussion. Okay.
Thanks, Irene. I'll try to get these centered. Good. So when I first looked at the concept document, the thing that struck me as a big difference in this topic from the question, for instance, of how molecules gain function is that one of the main themes here is actually, can we get experimental access to the emergence and the structure of evolutionary dynamics as a new kind of dynamical and organizational state? And one of the things I think we have the opportunity to do here that would be valuable is to not aggregate the way we think about evolution too much. If we think about the hallmarks that living systems have that distinguish them from non-living states, there are three things, all of which are important, but which are not the same. So living systems have the ability to form and maintain patterns that are away from equilibrium, but these patterns may or may not evolve in a Darwinian sense. There's a separate question of when the architecture of a system brings into existence some concept of individuality. And I agree with Andrew Ellington's comment that this is both an important but also a difficult thing to define or to conceptualize, and I'll come back to that in a minute. But even the emergence of a Darwinian dynamic is not the same as the emergence of the capacity for open-ended evolution. because for that you have to have the ability to maintain sufficient complexity that your space of ordered and maintainable states becomes effectively open-ended. And so to disaggregate these three processes is one of the things that I think there is beginning work in many areas of biology that we can draw on, and that hopefully this concept document can provide support for in various investigators' hands. So I will only talk about two things, the questions of individuality and modularity, and just give a couple of thoughts of things that are going on in present work that seem relevant to these. There are many ways of trying to get at the notion of individuality. Speaking very informally, the kinds of criteria we have in mind are that in whatever happens between the stages of reproduction, so generalizing what goes on in the physiology of an organism's lifetime or any assembly's lifetime, the components that are within an individual are interdependent on each other and connected to each other in one or more ways. Those among different units that we would call individual are autonomous, both in the way they work in ontogeny and also the way they are grouped or their fates are coupled when they're transmitted between generations. And all of these terms, of course, need to be worked out and defined in a system-specific way. One of the things that an individual is, as well as in dynamical terms, is in evolutionary terms, the set of components have to covary so that their inheritance together has fitness consequences for each of them. And these are things that have been formalized since the 1960s in the Price equation and Hamilton's rule for the consequences of covariance in relating one level of evolutionary transmission to another level at a higher or lower level of aggregation. And here, for my own preferences, I would not take a too materialistic view on this. What living systems do is create and solidify patterns and then transmit them and change them through time. And so in many cases, what we care about is the element that carries a pattern or the pattern itself, and how these things are handed down and how they covary. So to summarize, again informally, things that seem important to individuality, I think we could say that there's a notion of granularity within the independent units, and granularity meaning the character of constraints, the way the subsystems bear on each other is very different in a granular system than it is for a fluid system. And this is one of the things that makes biological evolutionary dynamics different from things like weather systems that are more diffuse and more continuous. And then the notion of shared fate, that characters that spent time living together wind up being transmitted together with greater than random probability. So where has work been done on this? When we formalize things as has been done in population genetic formalisms of evolutionary dynamics, it's easy to get into the habit of taking individuality for granted as a concept because we use it as an abstraction. But of course, in real material systems, we're not given these abstractions. Systems do whatever they do. And in living systems, one of the things that we see is that interdependence and then co-transmission can happen in a whole variety of ways that are structurally different and that can be opportunistically employed by a living system. So to kind of keep this slide from being all words, I cribbed a figure from a review by Matt Hearn and colleagues within the last year in Biological Reviews, trying to get a window on the complexity of individuality by looking at what they call the minor taxa, so the multicellular plants and animals that may have between a few and a few thousand species, but which have incredibly complicated forms of aggregation, development, and transmission. To come back to the slides here, what things do we know about the problem of individuality? We know, for one thing, that it may not be hard and fast. The notion of shared fate in transmission is often statistical. Richard Lewontin demonstrated this beautifully in the late 60s and early 70s with a variety of models of the maintenance of linkage disequilibrium in chromosomes as a thing that causes the gene to be a unit selection at the same time as the covariance in transmission of genes is also a unit of selection. So this, in some sense, cleaned up the problem of whether multi-level selection is essential. And this is sort of the first step in what takes us from additive genetic variance to the whole problem of modeling a developmental system. It's easy to think that individuality is limited to what we can see in genetic systems, but if we want to do small molecule experiments to get at these things, we're not necessarily going to be given the sophistication that modern genetic systems employ. And so it's important to realize that while we recognize individuality in development as having to do with which chromosomes are transmitted together, whether you have sexual and asexual generations that contribute to the generation of diversity, really what we're seeing is that the notion of individuality gives the environment a way to gate what forms of granularity and what forms of shared fate of phenotypic characters characterize physiology and then transmission. So let me switch away from individuality and toward modularity. This was another thing that was emphasized in the original concept document. Many authors have argued that you do not get hierarchical complexity of any kind unless you get it through modular intermediate stages. So Herbert Simon in the 60s and 70s had arguments pulled from a whole variety of domains of organization, from chemistry up through social systems. More recently, especially people who work in development and embryology have very many suites of rich examples here. So Mark Kirschner and John Gerhardt have written 2 long books as well as numerous scientific articles explaining where modularity is essential to both the preservation of functional robustness and the adaptability that that makes possible, because you can change things without breaking everything. One of the concepts that I think so to clean this and explain to you what this picture is, one of Herb Simon's examples was, of course, the Alexandrian Empire. This empire would not have been possible a few thousand years before, when you had disorganized small-scale tribes. The assembly of a large empire spanning, several time zones was only possible when you had large, stable state aggregates that could manage their own internal affairs. And though we may see this in geopolitics, once seen, we recognize that it's a principle of organization that applies ubiquitously. So the abstractions one can distill from many arguments, whether they come from Simon or Gerhardt and Kirschner or others, we can see that stability in a hierarchical system is a problem of control and of the complexity of controllers and the signals that mediate control. The problem of maintaining a stable architecture seems only solvable in systems that have the capacity to buffer errors at intermediate stages. And what modularity does is contain errors from propagating and provide some buffering within the modules. So these are general things. Can we show areas where they have been specifically important in biochemistry? This is an example from our own work. There was a paper in Physical Biology, for which Rogier Brackmann is the first author, looking at the origin of carbon fixation and its role in the larger architecture of metabolism. If you looked only at the network of carbon fixation and primary metabolism, you could immediately, from any number of network clustering algorithms, recognize that this network decomposes into sort of autonomous clusters. There are self-amplifying loops in the middle, which may involve the citric acid cycle or variants. There are one-carbon reduction strategies on cofactors that are separate from those. There's a whole network of sugar-phosphate reactions off to one side. At the same time as you could decompose this network topologically, you could realize, once it's decomposed, that the internal modules also have identity because they're specified by particular chemistry. So for instance, this one-carbon reduction has to do with specially adapted nitrogen-carbon-sulfur relations that mediate otherwise unstable one-carbon states. Things like the pentose phosphate cycle, whether it's used to make ribose or used to fix carbon, this is a whole network of sugar phosphate aldol reactions. So it's a specific chemical reaction being used over and over again. In the autocatalytic loops, what we find are patterns of growth and then fragmentation of molecules that are tied together by a certain set of thioester and iron-sulfur related cofactors. So we see that when modularity arises in systems like these, it may be given by the substrate, in this case the chemical and the energetic substrate, out of which you're making the system that you have to assemble. And this can be imprinted on the architecture of what you build. Does it have evolutionary consequences? Well, the surprising thing is that this is again from the same paper, and this is Rohir's figure. the entire history of carbon fixation actually shows a surprisingly serial sequence of innovations where you can recognize that elementary components, a part of a self-amplifying loop, a one-carbon reduction sequence, wind up being mixed and matched like blocks in a Lego set, and out of these you can assemble all of the known carbon fixation pathways that exist right now. The abstraction that we can pull from this assembly is that it simplifies the problem of innovation often to the creation of a single new type of chemical reaction. So you're not dealing with an irreducible complexity of having to innovate a variety of things. You innovate at the level of a single reaction, and then you reuse chemistry that you had before. The question of which innovations lead to viable outcomes then winds up being a question of network completion. So how can modules be assembled so that they are both functional and robust, meaning that they're sufficiently short, they're maintainable, they're insulated from each other? What we see at the level of small molecule chemistry then continues to hold at higher levels, including gene regulatory networks. So Doug Irwin and Eric Davidson and their various collaborators in both developmental regulatory networks and paleontology have written a series of papers over the last decade looking at the substructure in regulatory networks that's poorly captured in the way we think about both microevolution and macroevolution. And as Davidson and collaborators, many people actually working in developmental regulation, work out the structure of regulatory networks, it becomes clear that they're modularized and that the modules are heterogeneous. There can be modules deep and old that wind up accreting so many functional dependencies that they're no longer subject to evolutionary variation. They may have begun as simple functions and gradually been modularized, but as they get frozen out, they start to take on the same role as the kinds of constraints that we saw in chemistry in the previous slide. There then become secondary modules that depend on them that are more evolvable, but still the types of evolution that these things are capable of undergoing is limited because they wind up providing switches or inputs for downstream differentiation circuits. So these are the kind of substructures that make the difference between naive additive variance models and the kinds of real models we would like that give a rich picture of the way developmental architecture has evolutionary consequences. And the interesting thing is, at the same time as we can see these elaborations in developmental regulatory networks, we can see them in the early emergence of capabilities in deep paleontology, which then can wait a long time and then wind up undergoing efflorescences when they're triggered by environmental dependencies in many groups in parallel. And so this is what you start seeing in Cambrian diversification. So I think the story here is that all of the abstractions that we use when we're in a hurry are good for making formal models, but what we would like in the next round of experimentation is to get in small molecule systems, drawing on what we know throughout biology, at the fact that the progression from functions into things that become modularized or that were already partly modularized, to later constraints and to different classes of variation, we want to get at the structure of these and then understand how that defines evolutionary dynamics. So with this, I will stop. I'll turn over to Andrew, and this should be the next slide.
For one second, there's at least one question on the chat that maybe should just be dealt with now rather than going on. Andy, yes. Do you want to ask your question?
Is this Andy Ellington?
Looks like it. Okay, I'll just read it out because I can't hear him. Says, I'm uncomfortable with the notion of control, which seems a loaded term. Even regulation is subservient to fitness. All is subservient to fitness, including fitness itself. So there are networks that improve fitness and networks that don't. Whether or not there is control is irrelevant. Okay.
Well, I'm more than delighted to respond with a personal opinion, since we know that that's just a personal opinion and people will think what they think. When I use control, I want to use it in the way that engineers do in discipline of optimal control theory. There can be interactions between components, but the interactions may not be symmetric. And so the direction, for instance, in which a catalyst can influence a very large number of substrate reactions is not the same as the path through which the usefulness of those substrate reactions can feed back for selection on a catalyst. And so this can lead to a significant asymmetry, and it also leads to limitations in what you can stabilize. So there's a collection of classical theorems, many of which are connected to Ross-Ashby, which they're tautologies, but they're important tautologies, that when you have a hierarchical control system with an asymmetric relation in who can influence whom, you have the problem that the ability to stabilize the controlled within a certain domain is limited by the diversity of states that the controller can recognize and respond to. In living systems, it's also limited by the durability of memory and by the force that you can use to imprint memory onto controlled systems. And so I think in looking at hierarchical architecture, certainly in any modern life, but my own belief is even early as we start to get the rudiments of genetics and metabolic order out of geochemistry, I think that those asymmetric relations come in with catalysis and they become important right from the beginning. In terms of the relation to fitness, I think fitness is very much a two-edged sword. If you give me individuality that satisfies the axioms we take in classical population genetics, then I have the definition of fitness from Fisher and Price, and I know what we mean. It's a summary statistic which gives structure to evolution, and then we can approximate it with models in one or another way. Until we have reliable notions of individuality, it's not clear how we want to use the term fitness, and that itself is a thing that requires a lot of work. Initially, what we have is just messy dynamics. The question of whether that resolves into these concepts that make good conceptual distinctions, I think is hard work, which is a good place for theory and experiment to sort of mutually constrain each other. I hope that addresses the question sort of as it was intended, but I'm happy to, to come back even after the seminar and try to do better if it was not a good answer.
Sounded good to me. Why don't we move on to Andrew just for the sake of time?
The document has a series of excellent questions. It almost looks like an agenda for a generation or a couple of generations. What I really liked is that it brings together two concepts that are really different. The concept of evolution, which is a concept of sort of has its roots in genetics, and the concept of emergence, which is kind of self-organization idea. And just to remind people is that emergent properties arise when a number of simple agents generate complex behavior taken together. And they cannot be easily predicted by looking at those simple agents that come together. Another characteristic of emergent systems is that they arise from coupling between interactions at different levels. And this is actually one of the places where emergent properties meet evolution, because it's also a hallmark of evolution. Evolution, heritable variation come at the level of macromolecules. Fitness is at the level of the whole system, or like an organism. So we span different levels of complexity. And this kind of I understand that that's something that Eric also wanted to point out, is that the essence of trying to understand evolutionary phenomena is that we have to look at different levels of complexity simultaneously and understand how they interact. And perhaps that is not as strongly captured in the document as it currently stands, as it could be and probably should be. But what is really nice is that it stresses the fact that evolution and emergence are evolution coming from genetics, emergence coming from self-assembly are two sides of the same coin. And sides of the same coin are different, but at the same time they are inseparable. So they should be treated as such. And Well, I click at this down arrow and nothing happens. Oh, here it is. So the connection between emergence and evolution could be very subtle. That what you see in this slide in the upper left corner are vesicles, which are emergent systems. Yet these are emergent systems that, in simple circumstances, have nothing to do with genetics and nothing to do with evolution. Of course, in contemporary systems, membrane-forming material is generated biologically, and genetics comes into picture. At the bottom is something that I thought Eric might cover, but he didn't. And that's the question whether we can have whether we can have an emerging catalytic cycles and spontaneously, and there are diverse opinions on this. And Harold Morowitz thinks yes. Leslie Orgel thought no. But in the upper right corner, it's an ATPase molecule. And here you see this kind of interaction between genetics and emergence. There's obviously it's all genetically coded. Each protein each subunit that forms ATPase has been coded and evolved, but the whole molecule, which is essentially a small molecular machine, came to existence only because of intermolecular interactions, all through self-organization. So this was emergence of active transport was one of the key processes, if not the key processes, of creating life. And and one can say, okay, that's a product of evolution, and that's true. But one can say it's a product of emergence. It's also true. So it brings to light the fact that if we really want to understand processes of evolutionary innovation, which very often involve emergent properties, we have to couple those two issues: the genetic issue of evolution and issue of self-organization, which are not identical. Okay, so here is a closely related point that when we look at I showed examples of emergent properties like vesicles and or even some simple catalytic cycles, and they can kind of form complexity, lead to low complexity systems that are capable to evolve. And they have kind of low coupling, low complexity, and they lead to something that Martin Nowak called blunt evolution. It's only when we get high complexity and a coupling, then we get a robust and much faster evolution. But what is the relation between those two is not very well understood. And when we look at this, when we try to recreate some kind of robust evolution, it's very tough. And there is a long history, when we look at attempts, a long history of failure. And we don't entirely understand what's the connection between those two, how we get from one to another. That obviously people show that if you have autocatalytic cycles or systems which involves also mutual catalysis and so on, you can get robust evolution. But at the same time, there is a lot of questions how to create such systems. And what we don't quite yet understand is it a necessary condition? Yes, it is probably a sufficient condition. But is it a necessary condition? One can look at autocatalytic cycles as an example of something of a system with a strong bias. Now, we have other mechanisms to create strong biases. Can they lead to robust evolution? We don't know. And trying to bridge those first two boxes in this scheme is sort of one of the key questions in this thinking of evolution from the point of view of astrobiology, not necessarily from the point of view of other sciences, but from the point of view of astrobiology. And a part of this is also a role of neutral networks that we know that in principle they encourage innovation, but do they really exist? I mean, Irene will have a lot to say on this, and she looked at them, didn't find them, but it's possible that for more complex systems they might be there. And there are people like Andreas Wagner who are strong proponents of neutral networks. But then when you talk to microbiologists, they kind of say, well, what we see is really that neutral networks are not really the key to evolution. What we see are beneficial mutations primarily. So we can look at the problem from as a sort of before us as a two-component problem, something that is connected, but again it's different. Evolution is universal, so there are universal principles of evolution, and we should understand those universal principles. But what is on the left, on the right side, is historical reconstruction. I put them in quotes, sort of not to get misunderstood. It's just trying to say what actually happened at the origin of life, how evolution operated at the origin of life. And as far as we talk about universal principles, we can illuminate them, for example, through theoretical and computational methods, also through experimentation. And that might be a little inflammatory to say experimental A-life. But what I mean by that is that if you do experiments on let's say, evolving pieces of RNA in a test tube, you're doing experimental A-life. You're not doing historical reconstruction, unless, of course, you are very strongly committed to one specific scenario of the origin of life. Of course, the problem is that probably the earliest evolution was probably very messy and very unruly. And what are the systems? What are model systems at which we can try, even attempt any kind of reasonable experiment that tell us anything about historical reconstruction? There was in this draft document, there is this issue is raised, the issue of what are the right model systems, and I don't have any specific suggestions, but again, what I would like to stress is universal principles are for everybody. I mean, anybody who is interested in evolution is interested in finding universal principles. But this historical reconstructions, it's only astrobiologists that care. So this is a really astrobiology-motivated question. So finally, I would like to make a kind of a case for strict collaboration between experiment and theory, and I want to use my favorite scientist, Immanuel Kant, and who, here is this quote from him: "Thoughts without content are empty, intuitions without concepts are blind." And then he elaborates, and what he really means is that theories should be related to empirical evidence, and if they are not, they are useless. But at the same time, empirical evidence without theoretical generalizations really does not lead anywhere, does not lead to any deeper understanding of problems. And there is a lot to say about Kant's views on this, which are actually quite current today. But it's not the place to do that. But sort of ironically, this interactions between theory and experiment has been a little bit of a dancing lesson, kind of step forward, step back. That when the whole issue of molecular evolution started, let's say in the 60s and 70s, there was a very strong understanding that theory and experiment should go hand in hand, and work, let's say, of Peter Schuster and Manfred Eigen could be an example. Ironically, when very good experimental techniques for studying molecular evolution, like in vitro selection, were developed, then experimentalists stop listening to theorists, and theorists, to some extent, stop listening to experimentalists. And some of you might remember that just earlier this year, that there was at the Princeton meeting, that there was a theoretical talk, and Lauren Williams stood up and said, "Okay, show me the molecule." And I understand. I mean, I understand his point of view, but of course that tells us how deeply disconnected these things might be. And it comes from the view that expressed by one of my colleagues and very good scientists in the field, who said that origin of life is an experimental science. No, it's not. But and I'm really pleased to see people like, let's say, Irene in the new generation, who really kind of try to go back and say, okay, there are experiments and there is theory, and we have to view them as a single entity, and try to get this deeper understanding of this early evolutionary processes rooted both in theory and experiment. So we should very strongly encourage this kind of joint view, or perhaps even joint research in the future document on this theme. So thank you.
Thanks, Andrew. I just want to make one comment, which is that this document was originally entitled Experimental Studies of Evolutionary Pathways and Emergence. So sometimes in some of the places it hasn't been adjusted, but we took that out. We took out experimental studies in order to make it more inclusive. So please ignore any of the titles that read experimental studies of, and just consider it to be about the broader question of evolutionary pathways and emergence. Okay. So I know there are people who want to make comments. I know Uli for sure had some comments. Do you want to start it off? Just jump in.
And just so everyone knows, the phone lines are open. So if you were muted previously, just unmute and you can add a comment. And you can also start your webcam if you want to be seen while speaking. Thanks.
Uli, do you want to start?
Can you hear me now?
Okay. Oh, yes.
Hear me now?
Yes.
Yes.
Okay, excellent. Yeah, I have two comments. So I assume that I'm still commenting on the original document. Is that right? Because we had two presentations that were making a very deep background. So to the original document, you added the question, what is an individual? And I think that's very good. And I think that so in Eric's presentation, I think he went into the sub-question, what is an individual? And I think it's good to add this as a sub-question. And in one of Andy Ellington's earlier comments, he said, and I agree with that also what is evolvability should be a sub-question, at least to my understanding, that hard enough of a question that it could be studied independently. If this seems to be a concept that is very clear to you, then I suggest that if you agree on a definition of that, you could put it into the introduction of the Google document. And I think this is the only major comment I have. Yeah, this is the only major comment I have. I have quite a number of minor comments, and I put those later in the Google document. So what do you think about the sub-question, what is evolvability, or placing a definition into the summary?
Irene, we can't hear you, so you may have just muted yourself.
Oh, hello? Okay, sorry about that.
Ah, yes, that works so much better.
Okay. I know one of the sub-questions had been, what are metrics of evolvability? And that was basically saying, yeah, we don't know what it is or how to measure it. So we can put, I think it's a good idea to put in a separate question, what is evolvability? What does everybody else think?
Are we unmuted? I don't even know if we can be heard at this point.
I can hear you.
You can? Yep.
Okay, fine. This is Andy. I think it's a great idea. I think some of my early comments were just meant to do exactly what I think Eric really nicely did, which was set out a way to begin to discuss these concepts in a framework where we can not be held to a particular definition because these are hard things to define, but we can have discussions where if we agree upon common ground, theory and experimental, for how we evaluate terms, complexity can be evaluated by this, or individuality can be evaluated by that, then suddenly we have a way for theorists to play and experimentalists to play. So sure, put in a definition, but even more exciting is the notion of having common frameworks for discussion and defining those common frameworks as part of the roadmap.
Okay.
If several people...
Before we do anything procedural, can I add a comment to that of affirmation and extension? I didn't mention it because I couldn't find a way to present it on slides, but there's good statistical work coming from a whole variety of directions. People looking at causation, the ever-present community looking at network structure, people looking at information flows in hierarchical systems. Because so much of what we're going to do will need to be statistical, it strikes me that one of the good contributions of theoretical work in this area would be to try to draw on people's attempt to put some structure around these questions. And I think there is some good work that we can pull from there. So I agree very much, Andy, and would like to sort of say that's another thing to add to the mix.
So just coming back to, if anyone wants to ask a question or raise a comment, you're more than welcome as well to press the button up here, which shows raising your hand. I only mention that if sometimes people are reticent to start speaking because they're not sure if they're going to crash over the top of anyone else. So you're welcome to start speaking now. All the lines are open, but equally you can press the button. And don't forget you can type directly into the chat box. So other questions, observations?
Hi, this is Nick Hudd. Can you hear me?
Yes, we can. Hey, Nick.
So in some ways just some affirmations, but to emphasize, I think, the positive of keeping some push toward experiment leading and for sure being theory connected to that. The thing that I see is that Andrew brought up about self-organization and a connection to evolution. And I think that's important to think about that on a chemical level, right? What are the types of molecules that can actually recognize and self-organize? And that actually, that is a testable, right, question. And that is something that is probably a huge reason why, say, the peptides and nucleic acids, right? All of us, I think, would agree with that. And so that constrains chemical space, and that's a good thing to ask those questions. If not the molecules we have today, what is the space? And then touching on what Eric said about evolution, about open-ended evolution, I think that's a really important point because a lot of people will talk about something evolving, but it could evolve just to maximize what it does, and then I think it could be going to a dead end. And I think his point is really well made that there's so many different functions that had to evolve that we have to have, I think, pretty early on, materials that can do this. And again, it comes back to a chemical system level, or what type of systems allow you to have open-ended evolution quite early. And then finally, on to what Andy said, I think is important about not being held to a definition and just a cautionary note. That I know that when I talk to people about evolution, people will get hung up on, is this Darwinian evolution, is it not Darwinian evolution, right? And I don't think that we want people to get hung up on that because, again, there's kind of useful forms of evolution, right? An overarching thing is that maybe it's Darwinian early, maybe it's not, but there can be useful forms that allow us to emerge functions, multiple functions, and ones that don't. And one might be Darwinian, one might not, but we kind of have that bigger picture about what's going to get us to these more complex, useful functions, right? Not getting hung up on whether it's a collective or an individual that does it, right?
I think a comment I might have that is maybe a little too meta, I don't know, but that I was excited to hear about the introduction of probability into these frameworks and using probability as a tool to understand overlapping functionality and trying to understand how to relate fitness to the underlying system. Okay, not sure what that is. But in any event, the reason is in systems biology, and I'm sure many of you, including Andrew, have seen this, the notion of probabilistic gene function, of probabilistic interactions in networks has been completely enabling. I mean, it's transformed, in my view, how people view systems biology. And so as we move forward, being able to apply those concepts not just to life as it exists now, but life moving backwards and then at origins, establishing this really general framework for how to understand living systems in terms of network analysis and probability and fitness landscapes is the sort of thing that is so fundamentally important that NASA could take lead, but I know I've seen calls for proposals in similar veins from DARPA, from NSF. There may even be opportunities to partner with these other agencies to try and get, sort of interdisciplinary funding. Anyway, I'm babbling a little bit. I just think I'm very excited by what Andrew and Eric presented and think that this is really a transformative way to begin to set some of these questions down in the new roadmap.
Other questions or observations? And while you're thinking about it, just let me mention that we have now flipped the document so it is open for comment. So if you have any specific thoughts that you want to write directly into the document, you can just click on it from the front of the website. And the only thing we ask is it's helpful if you can log in to your Google account first, just so the authors can see who's writing what in case they need to come back and ask for any clarification. I know we have a few other people in the participant group who've been involved in the document. Aaron, is there anything you wanted to add or extend upon?
Sure. I just unmuted myself. So I think this has been a really great webinar. And one thing that I guess I want to add before we close is that I think that the last really interesting, especially because I've been thinking a lot about the semi-enzymatic hypothesis for the origin of metabolic pathways, and in particular how naturally occurring reactions that you wouldn't consider to be performed by an evolvable molecule can act in concert with evolvable agents as both as nodes of a sort of hybrid network. And I wonder if there's anything experimental that we could do. Certainly we could model it. I just wanted to put that out there.
Andy, so you don't have to call on anybody again. Can I say something? this is, of course, a personal preference, but it strikes me that in the enthusiasm for things that change, it's good not to lose sight of the fact that there can be a skeleton of things that never change. So there are things that evolve in the sense of being adaptable to circumstances that vary, and that can be a necessary part of robustness at the system level, the ability to flex. These need not be open-ended, but they can still be Darwinian in the sense that they can make use of the individual as the platform that carries memory and that changes the distribution of types. But if I look at the biosphere, I see patterns, whether they're co-evolutionary patterns in ecosystems or invariants in underlying metabolic chemistry and network topology, that are not changing now, and that underwent minimal change in the past, and that at the level of overall architecture look like they never have changed in anything that we can reconstruct. And if I think about the way reliable communication systems work, anytime you can preserve a variety of messages robustly for a long time, there's some system at the bottom of it that actually winds up being absolutely invariant. That's the sort of reference point to which everything returns. And so it would be nice, by my priorities, not to pass over opportunities to see where an invariant system can be a reference point for the complexity that then allows higher levels of variability. So they're all there at the same time.
And with that, unless anyone has any last thoughts or questions that they want to chip in, let me just say that the video of this webinar should be up on the website in the next few days. And I mention that just in case you have colleagues that you want to forward this to, so I'll be able to watch this. The document is open for commenting now, and I know that the team will really appreciate any feedback that you can give them to help strengthen the document and look at other directions they might explore. And I don't think we have any other takers. So thank you to our presenters. Thank you to all of the participants. And hopefully we will see many of you tomorrow. Thanks very much. Bye-bye.